This method can help answer key questions in the micro and nanofluidic research field, such as how effectively liquids are transported in narrow spaces. The main advantage of this technique is that cations and anions whose transport pathways are rectified by using an ion exchange membrane drive the electrohydrodynamic flow. Demonstrating the procedure will be Ayako Yano, an assistant professor of GUNMA University who graduated from our laboratory, and Fumika Nito, a PhD student from our laboratory.
First, adhere acrylic plates at both ends of a PTFE mold with a plastic adhesive, which will make slits in the reservoir to settle the bias electrodes. In a 50 ml tube, mix a silicone elastomer base and curing agent in a 10:1 ratio. Following this, settle liquid PDMS in a vacuum vessel and degas it using a rotary pump. Remove the tube from the vacuum vessel. Then pour the PDMS into a 40 by 50 by 24 mm cubed plastic vessel to mold the outer shape of the reservoir and place the reservoir mold in it. Bake the whole body of the liquid PDMS on a hot plate at 80 degrees Celsius for about 4 hours. After the bake, isolate the PDMS reservoir from the PTFE mold in the outer vessel by hand. Then make a slit across the center of the reservoir using a surgical knife. Using tweezers, set glass plates previously coated with a gold thin film at both ends of the reservoir to serve as the bias electrodes. Next,